Hello and welcome to Neuro Navigator. This is a mini-series focused on audition, and it's composed of human ear anatomy, hair cell transduction, major auditory pathways, and signal processing. Today's topic is human ear anatomy. The ear as a whole is a relatively delicate structure that undergoes a lot of wear and tear throughout a life. For today's demonstrations, we are going to be using this 3D model. So this outer external structure gather and focus sound energy, even helping to boost pressure for frequencies around 3 kilohertz. The shape of the outer ear and the pinna, which is this structure right here and here, is extremely important for localizing sound within the vertical plane. By reflecting the amplitude of sound waves depending on the way they hit the pinna, it creates a monaural cue for localization. So when you have a sound that comes like this, it's rebounded, it's reflected back to this ear canal right here. Through the ear canal, we get to the middle ear. This gray circle here represents the tympanic membrane or the eardrum. Connected to it and invisibly in this model are the ossicles. These are made up of the malleus, the incus, and the stapes. The middle ear is critical for impedance matching, and impedance is a medium's resistance to movement. It does this by focusing the force of the tympanic membrane onto the smaller oval window. If you didn't have this, nearly all sound wave energy would be lost by the. However, there is a regulation component to this too. The tensor tympani and the stapedius muscles counteract the ossicles' movement to reduce the amount of sound energy transmitted. And this does help to protect you from things like your own voice and also loud noises, but not sudden noises. The pressure waves modified in the middle ear are transformed into neural waves in the cochlea, which is represented by this green dot down here and does have a lot more structure that doesn't show up in this model. Within the cochlea is the basilar membrane, which vibrates in response to sound, or in other words, is displaced by pressure. The entire structure varies in width and flexibility, so different sections respond maximally to different types of stimulus. The base of the membrane is narrow and stiff and responds best to high-frequency sounds. The apex is wide and floppy, and it responds best to low-frequency sounds. If it helps to remember this tonotopic organization, remember that as you age, you aren't able to hear as many high-frequency sounds, which is usually due to damaged hair cells at the base of the membrane. The last anatomical structure we're going to be talking about today is in the cochlea, the organ of Corti. This structure is composed of both auditory supporting cells and mechanosensory hair cells, 1 row of inner hair cells and 3 rows of outer hair cells. It plays a role in auditory sensory transduction. Want to know more about auditory sensory transduction? Follow for more.